In this section of Chapter 25, we'll discuss SARS-CoV-2, a new coronavirus affecting humans and responsible for the pandemic of 2019, causing COVID or coronavirus disease 2019. The coronavirus SARS-CoV-2 was first detected in late 2019 in China, in a province of Wuhan. And in early 2020, the World Health Organization released a communique saying that SARS-CoV-2 is the responsible for the severe acute respiratory syndrome seen with the infection with this virus, and the disease was named COVID-19. By the end of January, COVID-19 was labeled as an emergency of international concern, and shortly after, it was considered a pandemic situation. Coronaviruses have been a concern for human health since the early 2000s, when we had SARS, which is a coronavirus, leaving an animal reservoir, probably in China as well, in the province of Guangdong, and infecting humans. SARS infect about 8,000 people with almost 800 deaths across 5 continents. And this epidemic of SARS died off few months after it had started. In 2012, we had another coronavirus of interest causing MERS. This Middle Eastern Respiratory Syndrome coronavirus emerged on the Arabian Peninsula, and it was from camels. The transference was from camels to humans. So the reservoir was a camel and then infecting humans. Very high mortality rate as well. And SARS-CoV-2 was first identified in 2019. So 3 coronaviruses had made the jump from the animal host into humans in the past 20 years. In addition to these 3 epidemic strains or species of coronavirus, we do have 4 low pathogenicity coronavirus that are endemic in humans. We'll see those coronaviruses popping up every flu season and causing flu-like symptoms with no pandemic ability. Like all the other coronaviruses, SARS-CoV-2 is an RNA single-strand positive virus with an envelope, and the envelope contains the spike proteins which give the name to this family of viruses. Corona means crown, so we see the spikes coming out of the envelope of the virus. Here we have listed the primary structural proteins on SARS-CoV-2, and the most important ones for our purpose at the moment are the spike proteins, or the S proteins. Those are the ones that the virus will use in order to enter the host cell. We also have an envelope, membrane, and nucleocapsid as part of the structure of the virus. The genome of the virus contains about 16 genes related to the biology of the virus itself. And interestingly, we were able to sequence the whole genome of the virus just a month after it has been reported, which was a pretty interesting advance in science. We believe coronaviruses became a human problem by jumping the barrier between species. That is one of the most accepted theories about the origin of SARS-CoV-2. And the transmission of these viruses is primarily through human-to-human contact. We can have, in some circumstances, animal-to-human with direct contact of animals to humans, and that is still under investigation. When SARS-CoV-2 was first being studied, we thought that droplets were the primary source of transmission. So we see here droplets, aerolized droplets from the respiratory tract being released by someone sneezing, coughing, or talking. Now we know also that this virus can be airborne. So even when the droplets have dried, these viruses can become airborne in a sort of dried droplet for an extended period of time, and that is a very efficient method of transmission. So those are two different things, and I need you to appreciate. Droplets, transmission through droplets is when the droplet contain water, and airborne is when even after the droplet had dried, we still have transmissible particles floating in the air for a period of time. Human gathering and proximity, physical proximity, will optimize transmission of this virus, which is primarily a respiratory virus. So the hands can also bring the virus, so the hands touching a contaminated surface and bringing the virus in contact with the body, as well as airborne or droplet transmission happens in population dense areas. Once the virus is transmitted from one infected individual to a healthy individual, the entry port could be the respiratory tract, and that's the most common one. We can also have the entry port in the mucosa of the eyes or oral cavity and esophagus. Once this virus enters the body, it will enter the host cell by binding to a particular protein in a surface of the host cell, and that is the angiotensin converting enzyme 2, or ACE2. The spike protein of the virus will bind to ACE2, which is present in a variety of cells in the host, and that will trigger the entry of the virus inside the cell. SARS-CoV-2 is primarily a virus of the respiratory system, but it will infect many other tissues throughout the body, causing different signs and symptoms of the infection. The mechanisms by which the viruses cause damage, as you will see, will have the same mechanisms that we described in other viral infections. First, the lytic cycle. Lytic cycle results in cell death, always. So once we have lytic cycles, we have cell death in the host, and that causes damage and sometimes losses of function. The immune system, in the best attempt to eliminate the pathogen, sometimes creates even more damage through cytokine storms, for example. So the immune system is the second major source of damage in COVID-2. And the last, and the one that we are still gathering more information about, since the SARS-CoV-2 use an important receptor to enter the body, the physiology of this receptor, once the virus has infected the body, change. So that can also cause damage. So to recap, lytic cycles, the immune system, and misfunction of the receptor used to enter the body can cause problems during COVID-19. ACE2 which is the receptor used by SARS-CoV-2 to enter the cells, have a normal function of controlling blood pressure, fluid balance, and some inflammatory response throughout the body. This receptor is present in different densities in different parts of the body, right? Such as lungs, kidney, heart, arteries, and gastrointestinal tract. The amount of ACE2 receptor is also controlled by the amount of testosterone in the body. So we will see a little bit of the difference between how this disease behaves in males versus in females. During the infection of SARS-CoV-2, we will see some disruption of normal cell functions, primarily associated with the functions of ACE2 also happening during the disease. On the next segment of this class, we'll discuss some more epidemiological aspects of COVID-19 as well as prevention methods.